One of the key steps in the discovery of new antibiotics is the hit validation and characterization stage, which helps confirm the early potential of promising compounds that can lead to the development of new antibacterial drugs. In the early drug discovery process, hit validation follows the initial hit identification stage, where chemical compounds with early activity against a bacteria or a specific biological target are identified. The initial hits are further evaluated, and the primary goal of hit validation is to confirm that the observed activity of a compound is genuine and not an artifact or false positive results. There are various strategies employed in hit validation that can be applied in a non-exclusive manner. For instance, you can run orthogonal assays where compounds are tested using assay formats that are different from the primary assays to confirm their activity and rule out potential assay interference or artifacts. Or you can also run counter screens to confirm a selectivity signal. Structure-activity relationship studies can also be performed, where analogues of the initial hits compounds are synthesized and tested to establish SAR. This approach will provide insight into the structural features responsible for the observed activity. Or you can also perform various cellular assays where compounds are tested in different cellular models to assess their activity in more details and in a more physiologically relevant environment. This is particularly the case if your primary assay screening was performed against an isolated molecular target or against a non-wild-type organism. At this point, it is also important to consider either a resynthesis or a purity check of your hit compounds to ensure that the activity is not due to an impurity in your samples or a degradation product. Hit validation is usually followed by a hit characterization phase, although in fact both phases can often to some extent be merged together. The validated hits are further characterized, and the manner and extent of the testing varies according to the therapeutic area or the organization standards. However, this phase often includes the evaluation of early physicochemical properties and in vitro ADME properties. Here again, the type of assay can vary, but general and microsomal stabilities of the compounds is often measured, along with their lipophilicity and solubility. A preliminary safety assessment is usually performed to evaluate potential cytotoxicity or off-target effects. Typically, the cytotoxicity is tested against at least one mammalian cell line, but the assay can be more specific or intensive to answer the needs of the project. If knowing the mechanism of action of your compound is necessary for your project to progress, this might also be the right time to investigate or confirm the mode of action of your compound. Additionally, and especially if you are working in infectious disease, you might want to test your compounds against a panel of strains of your species of interest or even across several species. This way, you will have a better idea of their activity spectrum. Overall, the hit validation and characterization phase is crucial on various levels. First, it allows the researcher to eliminate false positives. Indeed, the initial screening can sometimes yield false positive results due to various factors, such as compound interference or degradation or assay artifacts. The hit validation stage helps differentiate between those false positives and the compounds that are actually active to ensure that resources are focused on genuine and promising compounds. Hit validation and characterization also allows for a thorough assessment of potency and selectivity, or in other words, the toxicity towards human or mammalian cells. This information is essential for determining the potential therapeutic windows and for minimizing off-target effects. Next, and particularly in the context of limited resources, hit validation allows to prioritize the most promising compounds for further optimization and development, ensuring that the efforts are focused on the most viable candidates. And finally, with the insight you will have gained into the chemical and biological behavior of the compounds, as well as their potential liability, you will now have a solid foundation to plan the hit-to-lead phase that will come next. Because now you will have identified areas to focus on to make your compounds progress through the drug discovery process.